All right, good afternoon. The topic of this video is going to answer the question, can DNA be used to determine evolutionary history? So we learned last time that an evolutionary history really represents a family tree. So if we've got 4 species, A, B, C, and D, this shows their past. So these lines represent the passage of DNA from parent to offspring in a continuous lineage. Now, obviously, species B and C diverged from a common lineage fairly recently, right? So B and C are most closely related. B and D and C and D are still fairly closely related, but less so than B and C are. And finally, A is the least closely related to all 3. So what we have here is a family tree, and we've got species that are closely related, species that are distantly related, based on how recently they shared a common ancestor. More recent, more distant in the past. Now, in, say, the legal world, people and crime scientists can use DNA to determine relatedness. So, for example, if you have a kid and you don't think it's yours, you could have a DNA test done. This is based on the assumption that the more similar the DNA, the more closely related an individual is to you. So if you're interested in seeing whether or not a kid is your son, you're going to look for similar DNA. Why? Because you shared a very recent common ancestor, your parents. Okay? Someone who's not related to you is going to have very different DNA. So the question is, can we do this with species? Can we look at species DNA and determine their evolutionary history based on it? So to answer this question, we're going to rewind a couple million years, back to a time when Central America and South America were not connected. So this is a poorly drawn map, but it does show these 2 continents, Central America, South America, with a gap of ocean in between. This being the Pacific, this being the Caribbean. You guys probably know that nowadays there is a narrow strip of land called an isthmus containing the countries of Panama and Costa Rica, which connects Central and South America. What you may not know is that this isthmus is only a couple million years old, and that it was created by volcanic activity that built up a mound of new earth underneath the ocean that eventually broke the surface and created a land bridge between Central and South America. So the short of it is, millions of years ago, these oceans were connected, the Atlantic and the Pacific, by this little passage. So what I've shown right here is a shrimp population. Right? Shrimp are a marine invertebrate. They live in open water in anywhere from the surface zone down to several thousand feet. They are mobile in the water column, but they don't tend to migrate very far like larger whales and marine mammals do, and even some fish. So here's our shrimp population right here. And as you can see, we've got a Caribbean population that is linked to the Pacific population via this little water passage right here. Now, like any other organisms, shrimp have a genome. Right? They have genetic information with a sequence of A's, T's, G's, and C's. Right? Now, any given shrimp is probably going to be about 99.7% similar to any other given shrimp in its species. All right? Individual shrimp will have slight differences in their overall genome. There's their sequence of A's, T's, G's, and C's. However, they will be on the whole fairly similar to each other. The differences are comparable to the differences between humans, right? Humans all have pretty much the same genome, but our slight differences result in the differences in appearance and other traits that we observe between members of this class. Right? So that's what we've got for a shrimp population. Now you may also remember that genetic sequences obtain variation through the process of mutation, the big M word we've got here in kind of the glowing radius. Yeah. So mutations happen, and that's when there's errors in copying DNA, results in new DNA sequences, mutations, right? Now, usually in an interconnected shrimp population, a mutation that happens in one individual will be spread through interbreeding throughout the population. And because this is all one contiguous population, over many, many generations, this mutation that occurred here could spread and be found throughout the shrimp population. Right? Similarly, a mutation that happened here could spread be found throughout the shrimp population. So as a result of this, because of constant interbreeding between these members of the shrimp population, that will tend to smooth out the differences that might develop between this side and this side. Because we have gene flow between either side, over time the DNA sequence will change, but those differences will be shared, and as a whole, the shrimp population will remain, in general, fairly genetically similar to one another. So this is where it gets interesting now. We've got a contiguous population of shrimp, and we rewind back to 12 to 15 years ago, million years ago, excuse me, which is when most scientists think that the Panama Isthmus formed. Right? So let's go back to this time. Volcanoes erupt. They dump new lava, which builds up and eventually breaks the surface of the ocean. And all of a sudden, we have a land bridge linking Central and South America. Right? Good news for land animals, which can migrate to new spots now. But for these shrimp, there is no possible way that they can cross this land bridge. So what had been a contiguous population before is now two separate populations. The Pacific population, P, and the Caribbean population, C. So what happens now? Well, let's imagine that there's a mutation in the Pacific population. Boom, mutation. This mutation, through interbreeding, can be spread throughout the Pacific population, but it will not be shared with the Caribbean population. Why? Because shrimp from the Pacific one cannot make it across this land bridge to mate with those from the Caribbean population. What happened if there was a different mutation that occurred in the Caribbean population? Shown here in green. Different mutation, spread among individuals in the Caribbean population, not shared with individuals in the Pacific population. So this all harkens back to what you learned about gene flow with the M&Ms earlier in the year. When there's gene flow between populations, that smooths out differences between those populations. But when you create a geographic barrier resulting in reproductive isolation that prevents interbreeding, now these two populations are evolutionarily separate. They are no longer sharing their genetic information. And now they are prone to random forces, such as mutation. Right? By neutral theory, mutations will occur at random in these two populations. Because they're separate and not being shared, it's likely that eventually a group of mutations will develop that's very different and will lead to a very different character in this population than this one. And as a result, those mutations accumulate over time. Those populations will become more and more different. So that's all well and good. Obviously, when you separate two shrimp populations, they'll start to accrue differences through mutation. But how does this tell us about evolutionary relationships? It does so because of a hypothesis called the molecular clock hypothesis. Now, the molecular clock hypothesis is simple. It posits that mutations occur at a steady rate across time and across lineages. So in plainer language, mutations happen at a fairly steady rate, right? You'll have a mutation at a predictable rate across time. So let's apply this to our shrimp. What we have here is just a different diagram, a way of diagrammatically representing what happened to this shrimp population. So instead of showing the geographic scale, we're going to show them on a time and evolutionary distance scale now. Right? So here's our ancestor shrimp, right? Existing up until about 15 million years ago. Now, around the time period between 15 and 12 million years ago, the Isthmus of Panama formed. Right at this moment right here. And at that moment, we had the splitting of this ancestor shrimp population into a Pacific population and an Atlantic shrimp population. Right? So again, the key idea of the molecular clock is that mutations occur at a steady rate. So let's say that 100 mutations occur and become fixed in the population per million years. After 1 million years after divergence, we could expect to have 100 DNA differences between the Pacific and Atlantic shrimp. After 2 million years, we could expect to have another 100 come in, and we would have accumulated a total of 100 plus 100, 200 differences. After 3 million years, we would expect to have an accumulated total of 300 differences. After 4 million years, we would expect to have an accumulated total of another 100. That brings us to 400 differences, and so on and so forth. So that gives us a way, based on the number of DNA differences between two populations, to tell how long ago they diverged. Now we know already that these two populations, with a geographic barrier in between them, will start to accumulate genetic differences due to mutations. Their genomes will become different. But let's look right here. They've only been separated for about 1 million years at this point. Under the molecular clock hypothesis, a small amount of mutations will have built up. Because mutations happen at a steady rate, more and more mutations will build up the longer these two populations remain apart. So, if we were to go back in time and extract DNA from Pacific shrimp and Atlantic shrimp at this moment, say 1 million years after they diverged, their sequences would probably still be fairly similar, becoming more different, but still fairly similar. As we get another 1 million years on, their DNA sequences will be more different. 2 million years on, their DNA sequences will become even more different, and they will happen at the same rate because of this molecular clock, right? Another 1 million years further on, they will become even more different. So by the time we get to the present, they have accumulated differences to the point where they are much different than they were after they first diverged. So notice that because the number of DNA differences tell us how long ago two species diverged, we can use this to construct an evolutionary history. So let's say we've got 3 species, A, B, C, and D. Or sorry, A, B, and C, rather. We can compare A versus B. We can sequence their DNA and we can count the number of times that there's a difference in their DNA sequence. In this case, 45. We can also compare A versus C. Compare the number of differences in DNA, count each time there's a different base pair. And let's say that there are 112 differences between A and C. Finally, if we compare B versus C, we find 110 differences. So based on this right now, take a moment to answer for yourselves, which of these do you think are most closely related? Take a moment to answer. The answer, A and B. A and B have the fewest differences, which means they've probably been separate for the shortest amount of time. So if we were to draw an evolutionary tree with time on this scale, right, this being the present up here, and this being the distant past down here, we would show A and B to have a fairly recent common ancestor. What's our evidence for this grouping? The fact that they have few DNA differences. And based on the molecular clock hypothesis, fewer DNA differences means a shorter time that they've been apart. Short time they've been apart. Okay, now let's look at A and C. They have 112 differences. So A and C will obviously have a more distant common ancestor than A and B did. Right? A and C, they have a more distant common ancestor. Now finally, for the sake of solidarity, let's look at B versus C. B versus C has a very similar DNA difference profile to A and C, suggesting that B and C also shared a common ancestor about this far back. Right? So that's how you can use DNA differences to discern evolutionary relationships. This is a fairly recent field. Only in the last 40 or 50 years have we developed DNA sequencing technology. And it's been great because it's throwing a lot of traditional systematics and traditional groupings of organisms based on what was thought to be the evolutionary history on their head. And we'll look at an example of this with whales and with birds in class on Monday. Have a great weekend.